Hello everyone. We are here with another talk. This one is going to be in English. It'll be about one of my grad school work and education. So I hope you will find it interesting. I will start with that. I am assuming it'll take about 40 minutes or 50 minutes or maybe an hour. Uh, so let me start. So this is uh, this is about a course actually that I was taking when I was at UWM, UW Milwaukee. It, Actually, more than a course, it was like a talk, and I really enjoyed it. It was on Fridays around I think 5 p.m. Where everybody, the whole department would participate and come down, and they'll take the teas and biscuits and cookies, and they will bring whatever available, and usually the biology club would supply the tea and the coffee and everything, and then the cookies, and I loved it, to be honest. I mean, if you say tea and coffee, and there's sugar, and then there are interesting topics. It was really nice. So I hope you can read this. I tried to make it a little bigger. The colloquium, we called it. It was like it was like today's TED talks. Uh, People go and listen to interesting topics, and this was rather scientific and a bit longer. I mean, it would take around 55 minutes or an hour, and many of those actually were PhD dissertations and defenses and etc. The whole salon, the would be filled with everybody, the public could participate in. If I'm not mistaken, I mean everybody could come, and alumni, etc. So let me just and I took, it was one credit class, I believe, and I believe I took it every semester. Maybe it was mandatory, but like I said, it was 1 hour, but it was very interesting, and relaxing. And we had many interesting issues, topics. Let me start with this one. I mean, I think this was from year, I found it in my computer. I think year 07, 07, 2007. Let's just, this was the homework. I mean, we would listen to the talk and the speaker, and write about the topic, and the presentation, and then include some comments, on how it should be, and etc. And write a small question, if you were to ask. So I think, let me start with this one. I'm gonna read it in English. I mean, this is my handwriting, whatever I wrote it and submitted. This was this was a homework. So let me start with this one: environmental restoration and conservation. I guess this was the topic. I'm just gonna read very fast, and maybe I can skip some of the parts, but just so. Seminar was about an ecological restoration and conservation of aquatic systems in Wisconsin and Chicago area. Uh, Environmental issues were portrayed. Speaker noted the importance of sustainability and existing conflicts between economy, culture, and environment. And we still have, that the conflicts never end for some reason. Politics were reported to be crucial in conserving the environment, and Clean Water Act was explained. I think this was regarding Great Lakes. Biotic integrity was defined. That's important. Biotic integrity, because once you destroy that integrity, a lot of stuff can happen. And he noted that it was the most important criteria to evaluate aquatic systems. Biotic integrity. Biyolojik bütünlük in Turkish. Maybe somebody is listening in Turkish, but this will be in English. Speaker referred to some restoration projects in Chicago area. I do remember these talks actually. I remember all of them. And as I read, and I just my memory comes back, and maybe it is also relaxing for me too, because I loved those talks. Speaker referred to some restoration projects in Chicago area and noted some of the legal and environmental challenges that slowed and interrupted river restorations. I think he mentioned about Chicago rivers, and at the time they even changed the direction of the river and water basins. I do remember him talking about river restorations. He mentioned that some ecosystems were affected so badly that it creates hardship to analyze and plan restoration. I mean, if there is no ecosystem left, I mean, how are you going to restore it? So this is very important. I mean, you're trying to restore something, like you're trying to fix something. There needs to be something to fix. That isn't that true? I mean, how if you destroy everything, how are you going to fix it? Yeah, to fix something, at least, well, fixing means, just replacing one part, two part, restoring a function, and etc. But it has a limit, of if the degradation is so far, so much, then it is irreversible. You cannot restore it, and it says here it creates hardship to analyze and plan restoration. Finally, he talked about Racine area river projects and how public could be informed, and what systems and models have been used so far to explain issues to politicians and public. See, I mean, these. That's why I love these talks. I mean, the scientists have a duty to explain shit. I am sorry, to the politicians and the public. I mean, that is our job. Scientists need to explain, the importance and the problems to the politicians and to the public. If they don't, then how are you going to solve all these environmental problems or social problems? So public needs to know. I mean, there's just that comes down to this, so that you can explain stuff to politicians and public. But first, you need to study. So I made notes. I said the seminar was understandable, and I put note could be more interesting. I guess at the time maybe he was a bit boring. Presentation included a lot of images because I loved images. I don't like all text presentations. I wish I have text. I mean, I have images here, but but this is like a podcast, I consider this as a podcast. So, seminar was introduced with an interesting script. One could understand the subject matter and importance of the environment. So we were evaluating the talks at the same time. We were submitting these notes to the professor, who was in charge of, collecting the colloquium, homework and etc. Speaker did not have a monotone presentation, which is important because you put everybody to sleep if you have a monotone presentation. He displayed interesting images of his work subject matter. It was long. I would ask about the leading research centers in restoration and conservation science. That was my question, I guess. And this is another interesting. This is like I said. There was no there was no there was not a certain list for these talks. So they would arrange whoever they can find. They would come from like Texas and all over the country. They would come to make these talks. So I loved it. Everybody would come and and join the talks, and just and afterwards they would go and have more in-depth talk. Actually, they would continue their talk in UW building, Union Building. They would go there, and then if you had more, if it was in your field and you wanted to ask more questions, etc., and then you would go with them, and then they would have beer and stuff, and relax afterwards. So this was another talk. Small RNAs regulate gene expression in bacteria. So this is actually a good topic. Because due to their size, bacteria are small organisms. As we know, they are smaller than the eukaryotic cell. So that's why eukaryotic cell has many components that bacteria that doesn't have, because of the space. Like lots of signaling proteins, and all the cell surface proteins, and then signaling cascades and all this. And that is in fact to control DNA, or to control expression, especially gene expression. So in bacteria, things work differently. Even like the simple sugars, like like the glucose and or lactose, and if the glucose is not present in the environment, it changes gene expression. So it's very interesting, and we still have this model in our body. Our mitochondria still acts this way. Our mitochondria acts like a bacteria. So it's important to understand, and many of the molecules in our diet affects the mitochondria, because it is a similar system to bacteria. So if you understand bacteria, you understand your health better. Because like everything in our body is a result of energy. We are speaking, seeing, touching, and whatever, growing, protein, etc. It all requires ATP molecules and energy, and that is generated in mostly in mitochondria, and which is like a bacteria. So let me just quickly read. Seminar topic was very interesting. Small RNAs were defined and classified, and research history of such molecules was introduced. Small RNAs were found to be non-protein coding and not to be translated. So regulatory regions, regulatory RNAs. It is noted that mostly they were smaller than 350 nucleotides, smaller, smaller, because if they were protein coding, they would be a bit longer. Small RNAs are involved in RNA editing, modification, translation, regulation, protein translocation, control of translation, regulation of transcription. Until that day, at the high school, even at the college, they taught us that we have like tRNA, messenger RNA, and the ribosomal RNA. This was the first case actually I heard about small RNAs. Maybe this is your first time hearing about RNAs. And small RNAs, sRNA. Now everybody, with the pandemic, we learned about like mRNA, because they made a vaccine out of it, mRNA, messenger RNA. So messenger RNA, ribosomal RNA, and tRNA. So this is a new kind. If you haven't heard, this is a new kind, small RNA. So it's typed as sRNA. So I'm repeating, small RNAs are involved in RNA editing, modification, translation, regulation, protein translocation, control of translation, regulation of transcription. Speaker focused on her subject, sRNA, which was involved in editing RNA. Because RNA is the whole thing. RNA is the whole deal in the cell. It just translates into protein. She stressed that many of these molecules regulate gene expression and related to environmental responses. Her subject 6S RNA was abundant in E. coli. It was conserved in many conserved in many bacterial species. This specific RNA binds to RNA polymerase. That's just so very, very interesting. And inhibits translation by competing with DNA molecule. We all know that the RNA polymerase binds to the DNA and then it makes RNA. And if it cannot, then there's no gene expression. So it acts like a inhibitory element. SSRS gene mutation led to sRNA adult cells. These cells were not competitive and were able to grow on high pH environment. Normal cell growth causes the release of this specific RNA from RNA polymerase. She noted that nucleotide levels play an important role on determining the saturation of RNA polymerase. So like I said, these are important in understanding bacterial life. Like I said, I only knew 3 different kinds of RNAs, and this was the fourth one. So regulatory, small RNAs. Small RNAs regulate gene expression in bacteria. That is the take-home message. So let's continue. Seminar was very clear and much interesting. Presentation included a lot of images, figures. Seminar was introduced with an interesting topic. One could understand the subject matter and importance of this discovery. Discovery, that is the key word. That is a discovery. Because like I said, I only knew 3 at the time. Seminar gave overview of what speaker has discovered about these molecules. She outlined her research. Speaker did not have a monotone presentation. She displayed interesting images of her work and subject matter. It was enjoyable. I would ask whether regular mRNA, remember the pandemic, mRNA vaccine, and sRNA share any specific functions, other than like inhibiting or regulation, I guess that was the question I had in mind. Modeling species invasions. Now this is another talk. who Mac Isaac, that was I guess the presenter. Seminar was about prediction and modeling of invasion by non-native species. Invasion concepts were defined. Species poor, disturbed, and isolated habitats like islands were noted as relatively more susceptible to invasions. Speaker talked about characteristics of invasions such as it happens more in nutrient rich and as well as habitats that lack natural enemy. Features of successful invaders were defined as propagate pressure, strength of invasion. I know that characteristics of susceptible habitats were mentioned because today, as the habitats, get degraded, the risk of the risk of them getting, getting occupied by invasive species increases. So invasion risk increases as ecosystem degradation increases. Speaker explained several filters that invasive species have to pass in order to establish their founding population. That is, that makes sense. Human introduction is found to make these filters weaker and provides access to transportation to many invasive species, which normally cannot overcome by natural means. And we have, the speaker here, that's just basically talking about transportation. I mean, like, we have these huge ships, they have ballast waters, and then they're like, and taking tons of water, and then we have these planes and full of people, and then carrying all the germs from one continent to another. It just makes sense, and we lived this. I mean, this is year 2007, and then like after 12 years, guess what? We had pandemic, like Chinese mitten crab. Oh, very interesting. Chinese mitten crab was used as an example of invasion in Europe and North America. Oh, that's interesting. I wish it was only mitten crab. Ballast waters of ships carry this organism in between several ports along European coast and east coast of America. Ecologists designed a model to predict the vulnerable ports and harbors and possible inland migration and settling in inland lakes. As the transportation continues, so do the organisms. Just we help them. I mean, the invasive species and in agriculture, bugs, you name it, plants and weeds and bacteria, the fish. Nobody notices, but this is an ongoing problem ever. I mean, with the ships and with the transportation, with the intercontinental, business and trade, and then all these species that don't belong, to new continents or other places, because they don't have a predator there. I mean, so you have to protect the ecosystem. The ecosystem has a structure, and like I said at the beginning, if you lose that structure, say goodbye. And nothing is left behind. How are you going to restore it? I mean, if the whole thing is gone, Invasion tracking models were used to analyze these invasions. See, they were using models to analyze these invasions. Ecologists suggest that ballast water exchange during travel is an important measure to be taken during overseas transport. Like if there is a ship coming from Singapore, for example, once the ship reaches like a mid-ocean, region, it dumps the ballast water, so that it doesn't carry, whatever, it was getting from the port it departed. So it takes new ballast water from the ocean, the clean one, and then it continues the voyage or travel. That is so you have to change the water at least once or twice, or between ports. So that you don't carry unwanted stuff, unwanted organisms to new areas, etc. Hitchhikers or whatever you call it. Seminar was a very clear and interesting presentation. Did not include too many images, figures of ships and ports and ballast system. Seminar was introduced with fairly interesting topic. One could understand the subject matter, and etc. And then seminar gave overview of what speaker has found and predicted with the invasion model. Because if you have a working model, then you can predict stuff, and which is this is important in biological systems. I mean, you have to have a model. If it works, then you can predict upcoming upcoming invasions or upcoming pandemics. Speaker did not have a monotone presentation. He displayed some interesting images of subject matter. It was enjoyable. See, I told you, it was. I guess it was enjoyable. I would ask about specific effective methods to eliminate ballast water transport of invasive species. Specific methods, maybe like regarding fish or regarding smaller microscopic organisms, etc. I guess this is not important. Milwaukee sewage overflow. Even though Milwaukee had a big underground reservoir system, to contain rainwater and sewage water, so that they can, process it and send it to the plant and then purify it and then release it back to the lake. So this was regarding that, I think, from Freshwater Institute. The specific seminar subject was very interesting and important regarding Milwaukee area watershed and rain runoff to Lake Michigan. Because you have to protect the lakes and the water systems. I mean, very basic. And after every rain, they get polluted. I mean, this is no rocket science. Every country, on Earth, should know this. I mean, like basic. After the rain, all the shit in the cities gets washed into the lakes and seas. I mean, I'm sorry the language, but I have to make it clear. Agricultural pollution, that's the kind of shit I'm talking about. Agricultural pollution, sewage overflow, stormwater issues were main subject matters. Speaker introduced several important bacteria types that could cause risk in Milwaukee lakes and river systems, such as E. coli, which is a pathogen, and other pathogens and fecal coliforms. Hydrodynamic modeling was used to assess data. Year-wise analysis of rainfall, urban stormwater, and sewage overflow was studied. I mean, that you have to have people to study these things, so you can reach a conclusion. I mean, you have to have people studying the rain runoff and the sewage systems and etc. So you can make a meaningful decision for the city. I mean, you cannot just make up stuff or copy from other countries. Every city needs to study these subjects. Every university just needs to have a department studying these topics. Lake Michigan cannot survive after 6-8 hours. This was important, I think. According to water quality model and research, 90% of the bacteria that enters Lake Michigan cannot survive after 6-8 hours. I think that's depending on uh, the conditions. I guess river, harbor, and lake was extensively studied and sampled after rains and contamination. See, they were at least testing and sampling and doing something, just studying these contaminations due to runoff or other sorts of pollutions. Molecular tools have been used to track origin of bacterial species. Different bacterial species. They used molecular methods. Fecal pollution has been detected in the Kinnickinnic River due to a sewer leak. Beaches were also surveyed for bacterial contaminations. Gulls, seagulls were found to be effective scent polluters, and I mean that's not surprise. That's not surprise. When beaches were occupied by big flocks, I mean, like if there's like thousands of them, of course they are going to pollute the beach. I mean, that's not rocket science. Makes sense, and it is noted, and followed by rain runoff, and the rain washes all that stuff into the lake, and what happens? Several other tests were performed to gain more perspective of the water flow and rainwater dispersal. These are good studies, to help protect lakes and seas, And I said, seminar was very clear, much interesting. Presentation included a lot of images, figures, and seminar provided overview of what speaker has discovered about Milwaukee sewer area overflows, which is important, and she displayed interesting images of her work and Milwaukee Harbor and subject matter. It was enjoyable. I would ask whether other metropolitan sewer outflow data is relatively the same as Milwaukee area runoffs. Like I said, because if the Milwaukee is doing the homework regarding the sewage and the lakes and the runoffs and etc., what about other cities? I mean, they all need to be on the same page if they want a clean Lake Michigan, for example. This was important. And Milwaukee is doing, its part to my understanding. Microbial ecophysiology of carbon cycling. See all these different topics. I mean, so just a minute. Let me see how many minutes. It's 32 minutes so far. So let me just continue. Microbial ecophysiology and carbon cycling. See, this is about ecophysiology and carbon cycling, and this is the year 2007. Seminar was about ecosystem functioning and microorganisms. Speaker noted relations among soil environment and soil microbial community. Remember, like I am still today, like I am involved with mushrooms and etc. Speaker noted relations among soil environment and soil microbial community. Ecologists often neglect details of microbiota on ecosystem functioning. Bingo. That's why I am studying mushrooms today, because it's a soil. Is important. Soil is important. And it gets neglected, just simple. Ecologists often neglect details of microbiota and ecosystem functioning. Microorganisms are accepted as a black box, and processes are not completely analyzed except final outcome. It's just it's a cosmos, it's a microcosm. It's very simple. Size of the black box should be considered along details that we need, and the levels of those details should be considered. Speaker noted. Changes in the environment should change process rate and the microbial community structure and along resource availability. Carbon cycling. See again, like now we are saying the mushrooms are contributing to the carbon sequestration in the soil. you know, and see, it doesn't necessarily mention mushrooms, but it does talk about carbon cycling, and this just brings me, ideas about mushrooms. Carbon cycling was analyzed as a case study. Soil carbon can be found in great amounts to impact on atmosphere. Soil carbon. Again, we are going back to mushrooms. Like it doesn't mention, but in my mind, it does. How soil responds to temperature, because there is mycelium in the soil. There's microbial diversity in the soil. So we talk about an experiment that studies CO2. Even like preparing mushroom compost, like CO2 levels and acidity is important. CO2 temperature and soil relations. It showed that elevated temperatures declined CO2 respiratory rate. That's interesting. Substrate utilization will also varied with temperature. At high temperatures, I guess the soils the kind of stop functioning, and stop storing CO2 maybe. Seminar was very clear and was interesting. Presentation included a lot of images. Seminar was introduced with an interesting black box model. Again, in biology, all we have is models. I mean, in bacteria, there are models of regulation. In yeast, there are models of regulation. In ecosystems, and just you have to have a working model, and then you improve that model. You add things into it, you subtract or remove things from it, and that's how you understand. I mean, yes, we all have models in our brains. I mean, we have to have a working model, a working life model, so that we can work. It doesn't have to be the best one, but you have to have a working model in your brain, and it should be easy to control. I mean, like a Lego box, you have to be able to add things and remove things to your model of life, to your model of success, whatever you call it. And this is how scientists work. You have a model. If it works, fine. You try to improve it. If it doesn't, you change the model. Simple. And that's how people get their PhDs. Seminar provided overview of what speaker has discovered about the effects of temperature on microbiota and processes. And if the soil temperature keeps rising, what do you expect? I mean, things will stop. Basically, I mean, it'll become a desert. It just. And then it's important to model these things, so that you can predict an outcome. It makes sense. The speaker did not have a monotone presentation. She displayed interesting images of her work and answered interesting questions. It was enjoyable. I would ask about the largest scale soil experiments. See? Largest scale soil experiments that have been done so far in this field. Yes, everybody's doing experiments, but the scale, the how big are they? See, that was the question I had in mind at that time. See, this is an important one. By the way, this whole thing is like 9 pages long. I am still at the fourth page. Maybe I will make this a two-part presentation after this one, because maybe it is tiring. I will stop and then continue with a second one later. The effects of man-made nanoparticles and Daphnia magna. Daphnia magna, as you see, is called water flea. I mean, it's a crustacean. It lives in waters, and it's a good fishing bait for fish. I mean, you can actually culture it and feed your fish, etc. The effect of man-made particles. Well, remember, this is year 2007, and those nanoparticles were starting to show up in the market in those days, 2007. So this was an interesting topic. Sarah Lavan, good friend of mine, shout out to Sarah, very good friend of mine. She is such a good mother and a scientist and a lecturer and teacher, you name it. Shout out to Sarah Lavan. This was a PhD dissertation. I believe so. This was her PhD dissertation. And when I was a teacher, by the way, let me say this briefly. When I was a teacher, I brought one of my students. I wrote to UWM. I have a student and he wants to do a science project, and then they replied back and okay, with the freshwater science, Sarah would take the student, and then I would drive with my car and drop the student, and Sarah would take over, and she did some projects, I think copepods, with the copepods or the like, water microscopic organisms. She did some work with the eighth grader. I mean, so and then I think he loved it so much that I think he wanted to become a scientist. I mean, I was so amazed, and Sarah did that, So she helped with one of my students, and I can never forget it. She's such an angel. I mean, I cannot. What can I say? I mean, it's just she was doing her PhD work, and then she volunteered with a high school student in a project, in a science project. I mean, just what can I say? Good people exist. I trust in humanity. And let me continue. Speaker provided an outline for her presentation and introduced her subject matter in parts. Nanoparticle has been defined as particles in between 1 and 100 nanometer sizes. Increased use of nanoparticles threatens aquatic systems. Risks have to be assessed. This industrial substance has unique properties, such as high surface area. In her study, titanium dioxide was used in suspension as risk factor toxicity, effects of particle size. That's why, the rain runoff, all these industrial stuff on the streets, and then rain washes them into the seas and lakes. And what do you expect? You expect fish in that lake, in that sea, as a risk factor. As a risk factor toxicity, effects of particle size type were analyzed. Research showed that 10 nanometer particles showed higher mortality. Remember, the Daphnia, they are the bottom of the food chain. They are the fish food. Something happens to them, fish dies. I mean, it's just simple math. Daphnia magna, it's a crustacean. It's a small microscopic organism. Plankton, you call it whatever you call it. It just hops, that's why they call it water flea. It hops, The research showed that 10 nanometer particles, all these nanoparticles, showed higher mortality because it was the smallest, I guess. The smaller ones. Different types of nanoparticles were found to have different effects, such as carbon fluorine, heart rate, feeding appendage movement, hopping behavior, as well as tail movements were observed and quantified. I mean, you just, and it's an easy method. I would, when I was teaching, I would do the heart rate, which is rather easy. You trap the Daphnia magna with some cotton fibers, and then and then maybe with a stopwatch you measure the heart rate. I mean, you just drop certain liquids or pollutants and whatever you're testing, and then you can test the heart rate and then see if the stuff is good for the Daphnia or not. I mean, because stuff is in the lakes. Heart rate, feeding appendage movement, because they are filter feeders, they have to filter the water all the time, and if they are filtering the toxic materials, what do you expect? Hopping behavior as well as tail movements were observed and quantified. As a result, carbon particles were found more effective more than titanium particles, and caused risk vulnerable behavior to predators. And then, like I said, other fish, eat these, like minnows and stuff, eat the Daphnia. They're kind of Daphnia magna is the big one actually. You can see it with your eyes. I mean, it's just the biggest one, Daphnia magna. Daphnia, I think there's another one, Daphnia pulex, and Daphnia magna, or is it magma or magna? Maybe there's a typo there, but Daphnia magna should be Daphnia magna. As a result, carbon particles were found more effective more than titanium particles, and caused risky vulnerable behavior to predators. Seminar was very clear and very much interesting. Presentation included a lot of images, figures. Seminar was introduced with interesting images. One could understand the subject matter, importance of study. She summarized, outlined her research. Speaker did not have a monotone presentation. She displayed interesting images of her work and made jokes. It was enjoyable. As I see, she made jokes, and that's it. You have to be relaxed, so that you can work your brain. I mean, if you are relaxed, if you are in a relaxed mode of mind, state of mind, then your brain works better. It was enjoyable, I noted. And I asked whether other organisms like copepods exist in the same field of experiments, because copepods, she did the experiment with Daphnia magna, and then I asked copepods because I remember copepods from Balıkesir study, Manyas Lake. There's in Balıkesir we have a bird haven. Bird haven called the Manyas bird paradise, and then I remember we went there to collect some copepods, and then I knew them back in 2001, so I just asked about them. So, dear ladies and gentlemen, this is it for now. Let me see how many minutes have passed. Oops. Okay, I think 47 minutes, which is more than enough. Now that there's the time for questions, but there's nobody asking questions. So, by the way, here with all respect, I am now ending the live stream. Thank you very much. Please subscribe so that I can continue these kind of scientific talks and